Thank you, Hugo, for that generous introduction. I'm delighted to be here, and I'm particularly delighted that Denise will have a chance to talk after me this afternoon. So, needless to say, I'm delighted to just visit Stockholm again, and I thought I would give you an overview of a recent interest of my laboratory, and that is an interest in functional prions and their role in the persistence of memory storage. And let me put this into a little bit of a perspective for you. In the late 1970s and 1980s, Steenprusner began to develop the idea that there are conventional pathogenic prions. He developed the term prions as being based on infectious proteins. And he had the idea that these proteins exist in two conformational states, an inactive state and an active state. And the active state is capable of forming aggregates, and that aggregate becomes self-perpetuating. And one way it's self-perpetuating is it can act on the inactive form, cause it to convert to this active form, and the active form then can propagate forward. It can replicate itself, and it can replicate itself by also acting on the inactive form and causing it to convert into the active form. And that ends up in killing cells. In fact, in many cases, when it kills the cells, it's released by the cell that has died, taken up by normal cells, which in turn become diseased and killed by this. We've recently encountered an interesting variant of prions. They also exist in two functional states. They also are self-replicating, but they differ from pathogenic prions in two ways. One, it is not known what is responsible for the converting signal in pathogenic prions, but there is an actual physiological signal. Serotonin in the first example I'm going to show you, and glutamate in the second example I'm going to show you. By the way, there's a seat empty right there, which is my seat, and you're welcome to have that. At least for the next hour and a half. My lectures tend to be long. Each decade I add a few minutes. So number one, there's a physiological signal that converts the inactive form to the active form. And number two, the active form, which is self-perpetuating, is functional for the cell. In fact, the other conformation is usually a repressor function. The active form is an actual physiological function. And in what I'm going to talk about is in the initial state, it regulates local protein synthesis, important for the maintenance of memory storage. So I'm going to describe this to you in three biological contexts, going from very simple to more complex. I'm going to begin by telling you about work in Aplysia on simple forms of memory that Hugo referred to before. I'm going to focus on sensitization, not on habituation, where we use molecular and cell biology. Then I'm going to go to a more complex form of learning, associative learning, in the fly just very briefly, because this is largely other people's work. And then I'm going to come back to explicit memory storage involving the hippocampus. For people, places, and objects, I'm going to focus on places and speak about studies in the mouse. Let me begin with studies in Aplysia. As you can tell at the moment, this is not only a beautiful but a highly intelligent animal. This is exactly the sort of animal all of you would select to work if you wanted to study learning and memory. It has a terrific nervous system. It has a relatively small nervous system made up of most 20,000 neurons. These neurons are distributed in 10 ganglia, so each ganglion only has about 2,000 nerve cells. Moreover, for simple behaviors, the neural circuit is made up of less than 100 cells. Moreover, the cells are not only few in number, they're gigantic. They're the largest nerve cells in the animal kingdom. Before I became presbyopic, I could see them with my naked eye. These animals are not only beautiful, but they're highly recognized. And there are many people believe that it's the Aplysia rather than the people that should have gotten the Nobel Prize. And I'm going to describe to you the work that we've carried out now for some time, looking at this very simple reflex that the simple animal has. This, by the way, of those of you who don't know mollusks, is the head of the animal. This is its tail. It has an external respiratory organ called the gill, which is covered by a sheet of skin called the mantle shelf, which ends in a fleshy spout called the siphon. If you apply a tactile stimulus to the siphon, you get a brisk withdrawal of the gill, just like the withdrawal of a hand from a hard object. This simple reflex can be modified by 4 types of learning: sensitization, habituation, classical conditioning, operant conditioning. I'm going to focus on sensitization. Sensitization is a form of learned fear. So if you apply a noxious stimulus to the tail and scare the animal, you apply a tactile stimulus, it'll be much more profound than if you apply that stimulus in the absence of fear. And that enhancement of the reflex persists for a period ranging from minutes to days, depending on number of repetitions. The neural circuit of this behavior is quite simple. 24 sensory neurons that pick up directly from the siphon skin make connections to 6 motor neurons that move the gill. So it's direct monosynaptic connection that mediates most of the behavior. But there's also a side pathway of inhibitory and excitatory interneurons. What happens when you stimulate the tail? You activate modulatory systems, in which the most important is serotonergic. It acts on the sensory neuron, including its presynaptic terminal, and acts to strengthen the connections between the sensory neurons and the motor neurons. If you give a single tail shock, you get a transient strengthening, lasting minutes to hours, functional strengthening, due to covalent modifications of pre-existing proteins. But if you give repeated tail shocks, you activate gene expression and you have the growth of new synaptic connections. So insofar any of the students remember anything of this lecture, and I don't urge this on you, but insofar as you remember it, you will walk out of this lecture with a different brain than you walked into this lecture, and all without taking any drugs. Let me show you what happens at a molecular level. If you give a single tail shock, you activate serotonergic cells. They engage a receptor that engages an adenyl cyclase, increases the level of cyclic AMP, activates the cyclic AMP-dependent protein kinase, and it leads to a transient enhancement of transmitter release by increasing vesicle release from the presynaptic terminal. With repeated stimulation, the level of cyclic AMP stays up for a longer period of time. The catalytic subunit of the cyclic AMP-dependent protein kinase moves into the nucleus, as Roger Tsien and we were able to show in collaboration. In so doing, it recruits a MAP kinase. The MAP kinase removes an inhibitory constraint called CREB2 that allows the pKa to activate CREB1, transcriptional activator. It recruits the CREB binding protein and initiates a cascade of gene activation, leading to the turning on of late genes that give rise to the growth of new synaptic connections. And important kinesin transport system carries both messenger RNAs and proteins needed for synaptic plasticity down to the synapse. With great variations, this has turned out to be a general theme. Long-term memory involves transcription, almost invariably mediated by CREB alone or in combination with other transcription factors. And it leads to the growth of new synaptic connections. So although this has been of general biological interest, it's raised a very profound question in the biology of memory storage. If long-term memory involves transcription and therefore the nucleus, an organelle that is in principle in contact with every synapse of a neuron, does that mean every time you turn on a long-term process, you commit every single synapse of a neuron, 1,000 or more, to long-term memory? Or can you have a transcription-dependent process that is synapse-specific, that involves one set of synaptic connections and not the other? In order to explore that question, Kelsey Martin developed a new culture system. We'd always shown that because of the simplicity of the neural circuit, we could study in dissociated cell culture. But she now, by culturing individual sensory neurons and individual motor neurons and puffing on serotonin, we could simulate exactly what happens in the intact animal with learning. But now she carried it one step further. She developed a bifurcating culture with 2 motor neurons, so you have 2 separate branches, and you can see, can you produce long-term facilitation that is transcription dependent and one set of synapses and not the other? So she puffed on one puff of serotonin. She saw nice facilitation lasting minutes, specific here and not there. That we knew before. Short-term facilitation, which is just local, is synapse-specific. But then she gave 5 pulses of serotonin, turned on a long-term process that lasted several days. This required transcription. She could block it with an anti-CREB antibody injected into the cell body, but there was nothing there. So that indicated you can get a synapse-specific facilitation to this set of synapses and not there. But raise the question, what does it mean? Does that mean that the signal you send back to the nucleus now allows the transcription process only to be transported to this set of synapses and not that? Or is it the gene products are shipped to all the terminals, but only those that have been marked in a specific way can use those gene products productively? So Kelsey explored that. She gave a single pulse of serotonin to the other set of terminals. That usually produces facilitation lasting only minutes, short-term facilitation. But if it's done within a half hour, either simultaneously within a half hour of giving 5 pulses here, it captures the long-term process and produces in reduced form a long-term facilitation that lasts for days. And let me simply show you what that looks like. You give 5 pulses to one set of terminals. You produce a nice facilitation, change in synaptic strength, and an increase in number of varicosities, growth of new synaptic connections. If you now do the same thing again and you give a single pulse of serotonin, you capture in reduced form at the other set of terminals facilitation and growth, about half the size for the other set of terminals, clearly indicating that gene products are being shipped to the other side, but they need a marking signal. So Kelsey explored what the marking signal was. She repeated the experiment, 5 pulses here, 1 pulse of serotonin here, given together with an inhibitor of the cyclic AMP-dependent protein kinase. When you do that, you have no facilitation whatsoever. It blocks the facilitation completely. She now repeated the experiment. She did something more clever. She gave 5 pulses of serotonin here, 1 pulse here, together with an inhibitor of protein synthesis, either rapamycin or emmetine. And when she did that, she found something really quite extraordinary. She captured the long-term facilitation, had perfectly good facilitation and perfectly good growth, but it was not maintained. That it began to relax, so there was no longer there at 72 hours. What does that mean? It means that long-term facilitation has 2 components, an initiation component and a maintenance component. So there are 2 separate steps to it. And the maintenance component has something to it that requires protein synthesis. Let me just show you other examples of this. She could use rapamycin, which is a more restricted inhibitor. 5 pulses here, a single pulse here. Nice setting up of facilitation, including growth of new synaptic connections, and all of that regresses. Now she asked, is this also true for the site of initiation? Do you require this local protein synthesis at the site of initiation also? So you gave 5 pulses of serotonin, produced a much larger facilitation, much larger growth, but it was not maintained. So at both sites, you need a maintenance process. And let me just show you the quantification. 5 pulses here, a single pulse here, together with rapamycin. This is at the site of initiation, good facilitation, good growth. The site of capture, 24 hours, facilitation and growth. But at 72 hours, while it's maintained at the site of initiation, facilitation and growth, that doesn't see rapamycin, at the site of capture, you see that both the facilitation and the growth is blocked. So at this point, Kelsey Martin left my lab and I went into a funk, but fortunately, another wonderful postdoc came along, Kausik C, and he was fascinating about this process. He said, look, if you're going to send gene products to all the terminals, you have to be cautious because you want to make sure that these gene products are silent. Otherwise, they might be activated promiscuously, inappropriately, at inappropriate terminals. And one thing we don't want at the Karolinska is promiscuous activation inappropriately at inappropriate terminals. So he argued, you know, there are examples for this. There's an example in Xenopus oocytes where you have dormant messenger RNAs that are present, and they are dormant because they have a short poly-A tail. And there's a special protein called CPEB, cytoplasmic polydenylation element binding protein, that gets recruited. That binds to a CP element on these dormant messenger RNAs, and in the presence of CPEB, that recruits the cytoplasmic polydenylation machinery to lengthen the poly-A tail and activate these dormant messenger RNAs. So Kausik looked in Aplysia, and that activated messenger RNA can now be translated into proteins. So Kausik looked in Aplysia and found that the same developmental isoform that Joel Richter described is present there. But in addition, he found a novel isoform, never been encountered before, that is brain-specific. This was modulated by serotonin. So if you just give a single pulse of serotonin to one set of terminals, you increase the level of CPEB, and you block that with rapamycin. So this is a very good candidate for mediating that effect. So he tried to see what happens if you knock it out. So he knocked out, using an antisense oligonucleotide, CPEB, and he saw this is what 5 pulses of serotonin given without blocking CPEB. But if you block it, you have perfectly nice facilitation, but it's not maintained. Exactly what you see with inhibitive protein synthesis. He did this at 24 hours, he did this at 48 hours. So for some period of time, you need continuous synthesis of CPEB in order to do this. So this really looked like this was a candidate molecule that might be mediating the effect. So he examined it more carefully, and he saw, gee, he asked, what is responsible for keeping it going? And he saw it had a very peculiar N-terminal. What's unusual, there were a lot of glutamine and asparagines. About 50% of the residues of the first 160 amino acids are, in fact, these polar residues. Now, why is that odd? Because if you look at proteins like this, you see that in yeast, these proteins have prion-like properties. These are 2 classical yeast prions, and you see that the Aplysia CPEB falls right into this category. There's only about 45%. This is even more. Moreover, he noticed in the Drosophila, there's a Drosophila homolog, ORP2A, which I'm going to say something about in a moment, that also is enriched in these polar residues. So he wondered whether or not the Aplysia CPEB has prion-like properties. Now, the easiest way to examine prion-like properties is to go to yeast cells, because there are a whole sort of criteria for prions having been extensively studied in yeast. And so he began to explore this. First thing he did was to develop a reporter construct. You have a CP element to which CPEB can bind, fused to beta-gal. And when he put this into yeast cells, he found that many cells turn blue, as you might expect, indicating binding of the protein to the reporter. And that blue coloration was carried forward for generation to generation. But some cells appeared not to bind, and they were white, and they carried out that white phenotype from generation to generation. But there was interconversion at slow rates, but substantially higher than spontaneous mutation rates, between blue and white and blue. So if you took the blue cells and streaked them out, you saw an occasional white cell. If you took the white cells and streaked them out, you saw an occasional blue cell. So he wondered, what is the difference in conformation between the white cells and the blue cells? So he took CPEB, fused it to GFP, and when he looked at the white cells, he saw they were diffusely stained, as if they were soluble. When he looked at the blue cells, they were punctate, as if aggregates might be forming. To see whether these, in fact, were prion properties, he wanted to see whether, in addition to aggregate formation, they were heritable. They passed from one generation to generation. So he did a very nice experiment, which is classic for yeast, which is called cytoduction. And you take a strain of yeast in which you can get cytoplasmic fusion without nuclear fusion. And he took the blue cells and the white cells, and he fused them together. And after the fusion occurred, the blue protein moved into the white protein and caused the white protein to assume a blue conformation. So the soluble protein was turned into an aggregate protein as a result of the presence of aggregate proteins. So this fitted very much with the fact that it had prion-like properties. So we wondered, does this also apply to nerve cells? Does CPEB act like a prion in nerve cells? And we began to explore that. And after that, we said, is this conformation essential for perpetuation? So first question we asked is, what happens if you overexpress this? So if you overexpress this, fused to GFP, that is overexpressed CPEB in Aplysia, you see this very nice aggregate formation. Now in yeast, if you chop off the prion domain, you lose aggregate formation. So he chopped off the prion domain, and sure enough, he lost aggregate formation. Number two, he wondered, is it regulated by serotonin, which is what increases the level of the protein? And he looked at this, having expressed CPEB in Aplysia, fused to GFP, and after giving serotonin, you see a very nice increase in the level of the protein. And then he generated an antibody that was specific only to the aggregated form, did not recognize the soluble form. And he saw that indeed he was able to block long-term facilitation with an antibody that is specific to the aggregated form. So this is consistent with the fact that there are two conformations. The aggregated form is necessary for the perpetuation, and it is sensitive to inhibitors of protein synthesis. Now, how is this activated? How do you convert it from being a soluble form to an aggregated form? We've now studied this at some length, but I'm just going to give you the answer rather than taking you through the experiments. In the discussion later on, we can go through the detailed experiments. I showed you before that a single pulse of serotonin increases the level of the protein. That's absolutely essential because it's the increase of the level of the protein that allows the conversion to occur. You need to have a certain minimal concentration of the protein in order to get the switch going into self-perpetuating mode. But what is responsible for this increase? We've really found to our surprise that small RNAs, both microRNAs and piRNAs, have a major role in regulating memory storage. And at lunchtime, we'll discuss that. I'm not going to discuss that here. I'm just going to give you the result of this one set of experiments. We found that there's a specific microRNA. This is also present in the mouse, microRNA 22, that binds to and inhibits the messenger RNA for CPEB. What serotonin does is to remove that inhibitory constraint, increase the level of the protein, and once it's functionally induced in this way, it converts to the form, the active form that is capable to aggregation and self-perpetuation. So I have some beginning insight into it. And I think the reason I want to emphasize it, and I'll come to this later on, is this is a potentially explosive mechanism. So you want to regulate it at both ends, how you turn it on and how you turn it off. We have more insight in turning it on, as I'll show you later on. We don't as yet know, although we have some clues, how to turn it off. Let me give you the second example. This is work primarily carried out by Karolinska and by other people, but I'm just going to give you some beginning results that Karolinska started while he was still in our lab. He explored a more complex form of learning, looked at classical conditioning in the fly, and that was and he was interested in the homologue of CPEB in the fly, which is called ARB2A. And the first experiment we did, actually together, it was a very simple experiment. We wanted to take what happens if you put ARB2A into Aplysia neurons and you give serotonin. Can you convert it from a soluble form to an aggregated form? And we found that, in fact, the conversion pathway seems to be preserved. You could convert it. And then we did an actual behavioral experiment. Many of you are probably experienced with this. This is really asked the question of, why do you want a perpetuating mechanism? Well, one of the reasons you want it is you want to remember your first love experience for the rest of your life. Now, many of the gentlemen in the audience has probably had this experience, but if an inexperienced male is rejected by an experienced female, they remember this for the rest of their life. And this is even true for flies. So if you now explore what happens if you knock out RB2A in the fly, you find it in no way affects memory for this rejection experience, the short-term memory. But if you look at long-term memory, if you knock out one copy of the gene, you have a reduction in it. If you knock out both copies, you have complete loss of memory. And if you knock out the Prion domain, you have almost complete loss of memory. So in the fly, you can see that this also has a role. And this experiment was carried out by Barry Dixon. Karolinska has now continued with this and shown this very elegantly. What about the mouse? What about, real animals, mammals? Turns out that mouse and you have not only one CPEB, you have 4 isoforms of CPEB. But only one of them, CPEB3, has a Prion domain. We popped it into yeast, as we now routinely do on these things, and we found that it has classical properties of a Prion. And it is the only one. CPEB1 doesn't have these features, only CPEB3. It forms aggregates. These are SDS resistance. They're self-propagating, and they're heritable. We then asked the question, what happens if you put the mammalian protein into Aplysia neurons and you give serotonin? You see a very nice increase in level and in aggregation. Moreover, if you look at hippocampal neurons in culture, you see that if you give glutamate, the excitatory transmitter for LTP, you see aggregate formation in the processes. You not only increase the level of the protein, you increase the level of the aggregated form. What are the targets for CPEB3? It turns out they're targets that are important for long-term memory, GluR1, GluR2, and Pamilio. And you actually show this, which is quite fascinating. It really confirms what we saw in Aplysia. If you take hippocampal neurons and you overexpress CPEB3, you find that in the basal state, you actually depress the level of GluR1 and GluR2. So in the basal state, when it's not in its Prion state, it is a repressor of translation. If you're now activated with glutamate, you now increase the level of expression of GluR1 and GluR2. It now becomes an activator. Moreover, you find that when you activate it with glutamate or with learning, CPEB3 moves from a position in the dendrites to dendritic spines, where it becomes associated with polysomes. So you can use polysome markers, and you see the active form in the presence of glutamate has a significant amount of CPEB3 associated with ribosomes. And this simply shows you CPEB3 and markers, different markers for ribosomes. And you can see that they co-localize. And you can see this increased localization with polysomes in response to glutamate. So let me simply summarize in this cartoon our understanding of what is happening. In the basal state, in the cytosol, CPEB3 is inhibited. With activity, it translocates to the polysomes where it's capable of translating messenger RNAs necessary for the maintenance of learning and memory. So CPEB3 is upregulated by LTP, as I will show you, and it's important for really critical phases of memory storage. Let me simply show you this. If you produce LTP with 4 trains, you don't get it with 1 train, you see very nice upregulation of CPEB3 that lasts for several hours. Moreover, if you use a knockout of CPEB3, you selectively block the late phase of LTP. You can now begin to explore this in memory. And of course, in the mouse, we can really define different forms of memory and different phases of it. And I'm going to show you experiments that deal with consolidation, with maintenance, and with the phenomenon of reconsolidation. As you probably know, recently, a lot of emphasis has been brought to bear on a fascinating feature of memory storage. That is, if you form the memory and maintain it, in the first few days after maintenance, it's very sensitive to disruption. If you recall the memory, if something happens that causes you to recall the memory, it becomes very sensitive to distortion. So if you give inhibitive protein synthesis at that point, a much later time, you can abolish or severely interfere with it. And it's thought from an evolutionary and functional point of view, the reason this phase exists is because it allows you, under normal circumstances, to strengthen the memory if you want. It also gives rise to memory distortion, how you can incorporate alien information into the original memory. So for example, if you recall the memory again in a different context, you often incorporate aspects of the new context and distort the memory. And this is why testimonial of individual witnesses in court is a little bit tricky. So let me show you these experiments in sequence. Consolidation. These are controls. These are animals in which CPEB3 has been knocked out, and you can see that the learning of both of them is comparable. But by day 4, when consolidation begins to occur, you begin to see an impairment and become statistically significant in a probe trial after learning has been accomplished. So 24 hours later, you see that maintenance has been compromised. If you now do a classical maintenance experiment, you have both lines of mice expressing CPEB3, but you have a locked CPEB3 present in the brain, which you can move with a lentivirus CRE, so that after the animal has learned, this is the control, this is the mutant expressing the CPEB3 that is functional, but can be knocked out. We now knock it out, and we see when we give a probe trial, 2 weeks after we've injected the virus, throughout all of this period, the gene is there. Now we knock the gene out, you see that maintenance is severely interfered with. And finally, we can come to reconsolidation. We can ask the question, What happens if we give something equivalent to a protein synthesis inhibitor and do this with CPEB3? So simply to remind you what happens with a protein synthesis inhibitor, if you now train the animal and then you test it day or two after the training is completed, so 4 days of training, and the 5th day you give a probe child, and you now give Anisomycin, you see in the test 24 hours later that maintenance has been interfered with, okay? Reconsolidation has been significantly interfered with. And we can show that the same thing we do with Anisomycin, we can do with a selective knockout of CPEB3. Here's the reactivation trial, and this is how severely compromised reconsolidation is. Reconsolidation simply doesn't occur to a satisfactory degree if this is knocked out. So we have reason to believe that CPEB is important for maintenance, for consolidation, and for reconsolidation. Given the fact, as I mentioned to you before, this is so explosive, how is it regulated? This is a wonderful set of studies that Elias Papadopoulos has found. He had found earlier in the fly that a molecule called Neurolized, which is a ubiquitin ligase, enhances memory storage in Drosophila. And he came to my lab and he wanted to see whether similar things were involved in the mouse. I wasn't particularly interested in the experiment, but I was interested in Elias because he seemed very bright. And I hoped that sooner or later, by talking to people in my lab, maybe we'd get interested in CPEB3. Turns out that Neurolized is a key regulator of CPEB3. Ubiquitin ligase mediate post-translational modifications of proteins, and they can be used either for degradation, which is what usually happens, or for activation. He found that in the hippocampus, Neurolized is critical for the activation of CPEB3. The way it functions is that in normal circumstances, as I told you, CPEB3 is repressed, and it's a repressor of translation. With activity, it is activated and moves in relationship to ribosomes. And Neurolized normally turns over very rapidly. It itself ubiquitinated, but it is broken down by proteasomes. But in the presence of activity, this breakdown stops. Neurolized moves in relationship to CPEB3 and ubiquitinates it, and allows CPEB3 to become active to cytoplasmically polydenylate proteins that have been dormant and now can become active. So Neurolized is necessary for the activation of CPEB3. This allows the growth of new synaptic connections. So let me just show you some of the evidence in support of this. We showed with Neurolized and CPEB3 they're co-localized. One is in opposition to the other at presynaptic sites in the hippocampus, and they're in opposition to presynaptic sites. They're both postsynaptic, opposition to presynaptic sites in the hippocampus. And you use an antibody with one, you pull both of them down. In mice where you overexpress Neurolized, you increase the level of CPEB3, but not CPEB1 or CPEB4. So we have two lines in which we've overexpressed Neurolized, modest expression and severe expression. So we have a modest enhancement of CPEB3 in one line, a more significant expression in the other. And we have this in a tetracycline system. If we turn the gene off, this reverts to normal. We all also lead to a systematic increase in the targets of CPEB3, Pamilio and GLUA1 and GLUA2, and again in a systematic fashion. If we now block CPEB3, we block this increase, okay? So dominant negative reduces GLUA1, Pamilio, and tubulin. Mice overexpressing Neurolized show an enhancement of LTP, and the mice that overexpress Neurolized are smart as hell. So in most animals, it takes them 5 days to learn. Of course, the Neurolized mice do this almost on day 4, 3, and 4. So you see that it takes the wild-type mice a significant amount of time. And here in day 5, the Neurolized are much better than the wild-type. Only on day 12 are they equal. But then if you change the task at all by changing the position of the platform, this is a Morris maze, again, our boys Neurolized win out. If you overexpress Neurolized, you also have an increase in number of spines. So you have an increase in number of growth of synaptic connections. So that allowed us to do a very nice direct test. We could take CPEB3 and Neurolized and put it to hippocampal neurons in culture and express each of them alone and the two together and see how this affects growth, okay? So if you look at a control case, you have a certain number of protrusions, dendritic spines. If you express CPEB3 by itself, it's a repressor in the basal state, so you have a loss of dendritic processes, right? So protrusions are decreased. If you now express neuralized, which activates CPEB3, together with CPEB3, you have an increase in the growth of number of synaptic connections. If you knock out the prion domain of CPEB3, you lose that, despite the fact that neuralized is present. If you get rid of the ligase domain of neuralized and express it with CPEB3, no growth of synaptic connections. If you take CPEB3 by itself and monoubiquinate it, don't use neuralized anymore, you just express it monoubiquinated, boom, beautiful growth of synaptic connections. So this shows clearly that ubiquination of CPEB3, normally mediated by neuralized, but can be produced artificially, is critical for the growth of new synaptic connections. Now, I told the people in my lab that one example of a prion protein in Aplysia and mouse and Drosophila is a very nice biological curiosity. But two different examples, begins to approach a biological principle. So I said, no, where's the second example? So people began to look for second examples. And that's what I'm going to briefly describe to you. This is work that primarily carried out by Joe Raymond, but a number of people, including somebody from here, Philippe, whose work I'm going to refer to in a moment, has participated importantly in this study. And they found a novel example of a functional prion called TIA, T-cell intracellular antigen. And it is an RNA binding protein that is present in stress granules and that is involved in cellular stress, oxidative stress or glucocorticoids or something like that. Joe went on to show that it's also important in organismic stress, not just cellular stress, and that it's a protective factor for post-traumatic stress disorder. And it's a post-traumatic stress disorder protector only in females. It is a sex-specific protective factor in females for PTSD. So this is really a new gene-environment interaction. Here's a gene. By itself, it doesn't do anything. But if you put the animal into an environmental contingency, post-traumatic stress, that is capable of eliciting this, you see the effect of this gene dramatically. So I'll give you some examples. The first thing we did is what we always do. We popped it in the yeast, and it's an unbelievably good example of a prion protein. We have a paper coming out in Cell just on this alone. It has all the criteria as SDS resistance, it's self-propagating, it's heritable, and this simply shows you that it forms aggregates. These aggregates are resistant to SDS, but they are not resistant to boiling. You can see these stress granules in hippocampal neurons when you expose it to oxidative stress with arsenate, or you give it corticosterone. In both cases, it causes aggregate formation. You see this in the hippocampus. And if you produce animal to learn fear, you can see that this aggregate formation, which is SDS resistant, disappears. It also appears with corticosterone. So corticosterone simulates post-traumatic stress disorder. We now actually turned to a PTSD paradigm in the mouse. And that is we produce fear conditioning, and we exploited not only initially, but the characteristic feature of post-traumatic stress disorder is it really becomes apparent several weeks after the initial traumatic experience. And it becomes traumatic in a very generalized way. So if you become shocked, for example, in a context that has a particular physical conformation or particular odor associated with it, you respond with fear not only to the shock, but anything that reminds you of the shock, like the environment or the smell. And this is true here as well. So if you look at the learning process itself for contextual fears, which we used, using different kinds of tests, open field, elevated plus, conditioning odor, there's no difference in TIA between the wild type and the knockout. So you don't see any difference in the learning process per se. But if you look 3 to 4 weeks later in the memory process, looking at the females, you see they spend less time in the open field. Open field is an anxiety-producing situation because you don't know where you are. If you put them in elevated plus maze, they don't like to be in the open arm. They spend more time in the closed arm. And moreover, if you have a chamber, which we had, in which you train them and shock them in response in the context of a particular odor, they avoid that odor and go to the other side of the box where the odor is absent. This has no effect on males whatsoever. The wild type and the knockout are unaffected, while in the females you see dramatic effects. So this is sex-specific. Moreover, we see sex-specific alterations in LTP. So if you produce LTP in the mouse, you see a particular alteration in LTP. This is the naive mouse. This is the fear condition. There's a reduction in LTP. You also produce that if you give cortisone. Okay? This is present in the ventral hippocampus. Okay? In the female mouse, you lose the response, both to fear and to cortisol. No effect whatsoever. The male mouse shows no effect under any circumstances. Moreover, this is not just hippocampus. We also see this in the amygdala. How does this work? It turns out that TIA regulates the alternative splicing of the glucocorticoid receptor. There are two forms of it. There's the glucocorticoid alpha and glucocorticoid beta that are different splice forms of the glucocorticoid receptor. And it turns out that the beta isoform is more prevalent in the female mice. So particularly, it becomes more prevalent in the knockout of the female mice. The alpha form is unaffected. So this shows you the female wild type and knockout for the alpha isoform. The same with the male. There are no changes in wild type and knockout. If you look at the beta form, you see that in the male there is no change. In the female there's a change not only at the messenger RNA level, but in the protein level. So again, you have a sex-specific alteration in glucocorticoid receptor processing in which there's an increase in expression of the beta isoform, the glucocorticoid receptor beta. What happens if we knock this down so we don't allow this to occur? And this is simply an experiment in which we knock down almost by half this. We find that the effectiveness in turning on downstream genes is reduced. So one of the things that this does, this receptor, the glucocorticoid beta receptor, is to increase the level of phospho-CREB, and that is significantly reduced as a result of reducing the level of GR beta. So our thinking is that what we've identified is a regulator of alternative splicing, and that this really serves as a molecular node for coupling cellular stress to plasticity, learning, memory, and behavior. And that what this does is, by regulating the glucocorticoid beta signaling, it dramatically alters the physiology and the behavior of the animal. So let me simply conclude with one general idea and several directions we want to move. The TIA story is still evolving. I'm presenting to you sort of the early phase of it, but we're beginning to get an insight into the fact that prion-like mechanisms can appear in different contexts, and we expect that even further examples will emerge. And the simple point I want to make is that we have very good evidence that Aplysia CPEB, Drosophila ORP2b, the mammalian CPEB3, homologue of CPEB in Aplysia, and TIA represent a new class of prions. They differ from functional prions in two fundamental ways. One is there's a physiological signal that regulates it. Serotonin Aplysia, glutamate in mammalian brain, and fear in post-traumatic stress disorder. Moreover, the dominant form is self-perpetuating, as is the pathogenic form, but it has a physiological function. I just want to outline a number of future directions. The first one relates to TIA, we obviously want to see, does this have some clinical relevance? Can it be useful to understanding post-traumatic stress disorder, which is beginning to assume really epidemic proportions? With America involved in a number of wars, we in the United States feel it very keenly, but also post-traumatic stress is not limited to wartime. Rape, attack, all of these horrible events that happen to humankind produce post-traumatic stress disorder. And we're beginning to collaborate with epidemiologists to look at 3 cohorts. One is a cohort with this collaboration with Murray Stein, University of California, San Diego, that looks at before and after active duty deployment of military personnel that also has a significant percentage of female soldiers in it, so that will allow us to look at this, and we don't have any data yet, but this will be pending soon. With Myrna Weissman at Columbia, we're looking at a family-based anxiety and depression cohort that she's been carrying for a very long period of time. And the preliminary results, they indicate that there's a significant interaction between TIA-1 and SNPs for childhood adversity in the development of anxiety and depression. And finally, here in collaboration with the Karolinska Institute, with Katarina Laverbrat and Yvonne Forsell, they're examining a Swedish longitudinal population-based cohort study, and their preliminary results also indicate an interaction between TIA and SNPs for stressful life events and development of pathological anxiety. So these are early stages, but we really want to explore them further because they think we're likely to be very informative. And the final point is, as we move into this, we'd like to know, what is so characteristic about functional prions that distinguishes them from pathogenic prions? What is it that allows them to be regulated by signals? And what allows them to be distinguished from pathogenic prions? Are there some structural characteristics? So all the structural studies, and these are difficult proteins, both the pathogenic and the physiological, to carry structural studies on, but we've started this in collaboration with Wayne Henriksen. So conventional prions have a beta sheet structure, and that is how that flips into the beta sheet structure is not known because we don't know what triggers the pathogenic mechanism. It seems to be uncontrolled and almost a chance phenomenon. And beta sheets are not regulated. So this seems an atypical or unlikely candidate from functional prions. So we looked at other things, and Ferdinand Fumara was really important in having insights that maybe something like coil alpha helices could be a more likely candidate for mediated oligomerization that can be controlled. Because unlike beta sheets, coils can change their state in response to signals in the environment. He's carried out a number of experiments. We've published 2 papers on this to show that there are these Q-enriched prion domains of CPEB that have the predicted pattern of the hydrophobic residue similar to that you see in coils. If you destabilize that, you lose the capability for aggregate formation in Aplysia neurons. So this and other information has made us think that it's likely that coils would be involved. Luana Ferrettini and Ferdinando Fumara have actually developed a model whereby this could provide a very nice scaffolding that allows binding through the prion domain of aggregates of these functional prion proteins, which could serve as a lattice for translating a number of different messenger RNAs in a coordinated fashion in order to meet either the demands of TIA or the demands of CPEB3. So let me end by simply telling you this is work that's been carried out for a decade or so. And it began, as I pointed out, with Kelsey Martin and Cal6C. These are all have left my lab and are happily ensconced elsewhere. Also, as Ferdinando Fumara, it's continued with work in my lab right now by Luana, Elias, Pierre, Joe, and Bettina. And I've been very fortunate to have Philippe Melas in my lab, who's not only carrying work on TIA, but also got us involved in the epidemiological work. And Martin Schalling, who's in the audience, has been a major contributor to that. I've also been very fortunate in having other collaborators, Susan Lindquist, who helped us with the early study of yeast, Wayne Henriksen and Ann McDermott, who's been helping us with the structural studies, and Irina Derkash and Myrna Weissman, who've been involved in yeast studies on the one hand and epidemiological studies on the other. And these people were also involved in the epidemiological studies I've referred to. So thank you very much. I'll be glad to take any questions you have.